Autophagy dysfunction is a common thread connecting the two seemingly divergent outcomes of aging: neurodegeneration and cancer. While these conditions manifest in different organs and appear to follow opposing trajectories, one involving cellular loss, the other uncontrolled growth, they share a critical underlying defect: the inability to maintain cellular homeostasis through effective self-cleaning. In both neurodegenerative diseases and cancer, impaired autophagy plays a pivotal role in tipping the balance from healthy aging toward pathology. In the aging brain, autophagy serves as an essential mechanism for removing damaged proteins, defective mitochondria, and other intracellular waste that accumulates over time. Neurons, unlike many other cell types, are largely non-dividing and highly metabolically active. They rely heavily on autophagy to maintain long-term function and integrity. When this process begins to fail, either due to genetic factors, metabolic stress, or simply the passage of time, cellular debris begins to pile up, interfering with neuronal communication and triggering toxic cascades. In Alzheimer's disease, for example, impaired autophagy contributes to the accumulation of beta-amyloid plaques and tau tangles, hallmark features of the condition. Similarly, in Parkinson's disease, defective autophagy prevents the clearance of damaged mitochondria and misfolded alpha-synuclein, promoting oxidative stress and eventual neuron death. The consequence of this failure is not just cellular dysfunction, but system-wide degeneration. Once autophagy is compromised, the brain becomes increasingly vulnerable to inflammation, oxidative damage, and synaptic loss. Cognitive decline, motor impairment, and eventually full-blown neurodegeneration follow. Therapeutic efforts to restore autophagy in these contexts, using agents like rapamycin, spermidine, or caloric restriction mimetics, are being actively investigated, and some early findings suggest potential for slowing or even partially reversing the progression of neurodegenerative disorders. Meanwhile, in cancer, the narrative is more complex. Here, too, autophagy dysfunction plays a central role, but with different outcomes depending on timing and cellular context. In early tumorigenesis, impaired autophagy removes a key line of defense against DNA damage, oxidative stress, and cellular senescence. Cells accumulate mutations unchecked, and the absence of autophagy removes the braking system that would normally push them into self-destruction or immune recognition. This makes impaired autophagy a cancer-promoting factor in the early stages of disease. However, as tumors become established, they often exploit autophagy to survive. In nutrient-deprived or hypoxic environments, tumor cells can reactivate autophagy to scavenge internal resources and resist cell death. This is particularly common in solid tumors where parts of the tissue receive inadequate blood supply. In this phase, autophagy supports tumor growth and resistance to therapy, and some cancers even become addicted to autophagy as a metabolic strategy. Thus, depending on the stage and type of cancer, autophagy dysfunction can either promote or suppress disease progression. What links neurodegeneration and cancer is the breakdown in cellular quality control mechanisms, of which autophagy is a cornerstone. In neurodegenerative diseases, the failure to remove toxic proteins and organelles leads to cellular exhaustion and death. In cancer, the same failure, particularly early on, permits the accumulation of mutations and cellular immortality. Both conditions reflect different forms of biological chaos, one through loss, the other through excess. Aging amplifies these vulnerabilities. As autophagy becomes less efficient with age, neurons lose their protective barrier against protein aggregation, and tissues across the body become more susceptible to the kind of damage that initiates cancer. The shared foundation of autophagy dysfunction suggests that interventions targeting this pathway may offer dual benefits: neuroprotection and cancer prevention. However, this comes with a major caveat. Stimulating autophagy too broadly could also help tumors survive once they've emerged, while blocking it in advanced disease could risk worsening neurodegeneration in other tissues. This complex relationship reinforces the need for precision in therapeutic design. It's not enough to activate or inhibit autophagy universally. The timing, tissue type, and disease state must all be considered. What protects the aging brain might fuel a growing tumor. What slows a tumor's advance might accelerate cognitive decline. Understanding the dual roles of autophagy in aging-related disease is not just a scientific challenge, but a medical imperative, requiring personalized strategies that respect the paradoxes embedded in our own biology.